Today we're going to cover Chapter 15 on seedless vascular plants. Now starting us off, we're going to have William Henry Lang. He's going to be a British botanist and phlebotomist paleobotanist that's going to be born in 1874 and dies in 1960. His untimely death of his father is going to be prompted by the family relocating to a Scottish rural bridge of Ware. And that's where he's going to become interested in botany and natural history. After graduating from the University of Glasgow in 1894 with a degree in botany and zoology, he's going to receive his medical degree in 1895, although he's never going to practice medicine. He's accepted a position in 1895 in the Botanical Department of the University of Glasgow, where he's going to focus on the development of morphology of non-vascular plants. So while studying fern species abroad, he received his doctorate from the University of Glasgow in 1900, leading to his appointment as Professor of Botany in 1909 at the University of Manchester in England. Research is then going to shift. He's going to become more interested in the study of things like plant fossils, so he shifts that to paleobotany. And so he's going to look at the plant fossil morphology in 1915 with the Cooksonia, a fossil that's believed to represent early vascular plants. As his most notable find, he's going to go in and look at this. He's going to receive awards and honors that include the election to the Royal Society in 1911, 2 honorary doctorates in 1932 and 1942, and he becomes the recipient of the Royal Medal in 1931 and also receives the Linnean Gold Medal in 1956. So with the evolutionary history of seedless vascular plants, we're going to have bryophytes that are going to dominate the terrestrial landscape during the Middle Ordovician or Early Silurian due to evolutionary innovations that are going to permit the colonization and proliferation. But, according to fossil record, plants are going to begin to start to transform and transition from non-vascular to vascular plants sometime around 440 million years ago. Several pieces of evidence are going to support this evolution. One is going to begin around 440 million years ago. We're going to see the appearance of what we call trilete. So we got these trilete cryptospores that are starting to form. Also, we're going to see vascular tissue of xylem and phloem coming about and lignin. So as the abundance and complexity of these structures continue to evolve throughout the Silurian and Devonian periods, significant modifications in the alteration of generations life cycle, nutrients transportation, and mechanical support are going to begin to appear. So the earliest representative of vascular plants are going to be the rhinophytes. This is going to be a group of plants from 425 million years ago that had a trilite spore and vascularized stems but lacked roots and leaves. An example of early fossilized rhinophytes are going to be the Cooksonia that was discovered by William Henry Lang and Isabel Cookson in 1934. It was later described and named by Lang in 1937. Now here we have the Baragawanthia, and that is going to be a large vascular plant with a trilite spore that's going to have a thick stem and first to have vascularized leaves. This fossil was described by Lang and Cookson in 1935. So the increase in leaf complexity and vascularity in the early vascular plants is going to continue throughout the Denovian and Carboniferous periods, a series of adaptations that are believed to maximize photosynthesis. Now we're going to have true vascularized roots, with the mycorrhizae appearing in the fossil records around 407 million years ago as a simple rhizoid-based system, while larger penetrating root systems are going to appear around 390 million years ago. Early vascular plants are going to begin as small herbaceous plants due to their diversification, advancement of vascular tissue, and refined reproductive potential. They're going to then create large, extensive forests, which are going to dominate the terrestrial landscape throughout the Carboniferous and the Permian periods. Today, the nearly 12,000 representatives of seedless vascular plants are much smaller than their ancestors. However, they exhibit many ancestral traits including trilate spores and vascularized tissues. As always, we have to talk about the scientists who are going to contribute during this period and during the evolution of these plants. So first we're going to talk about Carolus Linnaeus in 1735. He classified seedless vascular plants as cryptogams, flowerless seedless plants that are going to be further divided among the mosses and the ferns. Next we have Pelissat de Buevos in 1803 to 1805. He's going to be the first to classify quillworts, club mosses, and his new genre for spike mosses, which is going to be Selaginellia. That's going to be a collective group that he named the Lycophytes. Third on this list is going to be Olaf Schwartz in 1806. He's going to revise the classification system of ferns, including a new genus of whisk ferns called Solithium. Other important botanists during this time are going to include Nicasio Auguste Desvaux in 1827, Carl Bourgeois Purcell, 1836 to 1852, Antoine Laurent Apollinaire Free in 1850 to 1852, and Sir William Jackson Hooker, 1844 to 1864. They proposed more modern classification systems for ferns throughout the 19th century, which further differentiate the Pterophytes, which are going to be your lycophytes and ferns, and distinct groups of plants. Here you see Frederick O. Bauer, 1928. He's going to be the first to propose a phylogenetic classification system just for ferns. Despite the instability of phylogenetic classification systems thereafter due to shifts in interpretation of plant collections and classification approaches, more influential classification systems were developed by the 1940s as better classification practices were implemented. By the 1990s, the use of molecular studies continued to improve the phylogenetic classification system of these pteridophytes. So the use of molecular data is going to help to improve the phylogenetic classification system by revealing evolutionary relationships and the dichotomy among the tracheophytes, the lycophytes, and the euphylophytes. These are going to be groups that are going to include the monolithites and the lignophytes. So a new classification system for the monolithites is going to be developed using a combination of molecular and morphological data in 2006. In 2016, a community of botanists developed a new classification system for all pteridophytes using recent molecular data. So although some botanists were hopeful this classification system would bring stability, others have called it unpopular and confusing due to the excessive splitting of the genre and introduction of new nomenclature, which they do not influence future classification systems here. So current classification systems are going to include the lycophytes, which are going to be your quillworts, club mosses, and spike mosses, which are going to be considered sister groups to the euphylophytes. The euphylophytes are going to be a monolithyte, which is going to be horsetails, whisk ferns, and ferns, a sister group to the lignophytes. These are going to be recent studies that are going to suggest that horsetails are going to be sister groups to all other monolithites. Pteridophytes are going to be multicellular eukaryotic plants. These are going to be classified as tracheophytes because they have specialized nutrient-conducting vessels that are composed of two types of vascular tissue that comprise roots, stems, and leaves. The first type of vascular tissue is the xylem. That's composed of thin-walled parenchymal cells that are going to store nutrients and thick, non-living sclerenchymal cells that are going to be reinforced with lignin. These are going to transport water and minerals. So think of xylem and water and minerals. Phloem is going to be your second type. It's composed of modified parenchymal cells and supportive sclerenchymal cells. These are going to help to transport sugars and other organic nutrients. Now here we can see pictures of these vascular tissues and artist renditions or representations. So with the xylem, remember that's going to transport your water and minerals through vessel elements. So these vessels that you see here in pink are going to show how you're going to have these little sieve systems or these little slits. They're going to be perforation plates, and that's going to allow the fluid to flow upwards but help to keep it flowing in one direction. And you see the parenchymal cells, they're going to be supportive cells around it. You also see that they have tracheids that are going to be on the sides there. Now coming on over to the phloem, that's going to transport sugars and other items that are needed. And we see that here they're going to also have a sieve tube element that is going to force it to come through these openings or these circles there. So with the pteridophytes, you're going to have roots that are going to help to anchor and absorb nutrients from the soil and develop from an underground stem known as a rhizome. Rhizomes also give rise to stems, which are going to help to support the plant as it grows upright and provides an attachment point for leaves. Now you're going to have two types of leaves, which are going to act as the plant's primary photosynthetic organ. You have microphylls. These are going to be small leaves with a single strand of vascular tissue. That's going to be found in the lycophytes. Then you could have the megaphylls. These are going to be large leaves with multiple branches of vascular tissue. These are going to be found in the manilophytes. Alternation of generation life cycles of the pteridophytes are going to include a small gametophyte and a large dominant sporophyte, both with protective cuticles around them. So remember, the cuticle is going to be a waxy-like structure that helps to prevent desiccation or drying out. So with the gametophyte, it produces gametes and is going to carry out the photosynthesis, but it does not contain vascular tissue or the stomata. Remember those openings. The sporophyte is going to have vascular tissue, it has stomata, and it's going to be responsible for producing spores. Now about 85% of pteridophytes are going to have mycorrhizae found in their species. This is going to help plants with nutrient acquisition, protection from pathogens, and improving tolerance to environmental stress, and plant provides photosynthetic carbon and sugars to the fungi in return. Studies have revealed that about 68% of pteridophytes and less than 10% of their total diversity have formed symbiotic associations with mycorrhizae. Reliance upon mycorrhizae is going to be found predominantly in the pteridophytes in nutrient-deficient substrates and extreme habitats that are going to be exposed to high wind and temperatures. Association has also been found to decrease with increased elevation due to low temperatures influence slow fungal growth and insufficient cost to plant based upon the return that they would have received, so it's not worth it in these higher elevations. Nutrient-rich substrates are going to accommodate non-mycorrhizal pteridophytes, and that's going to help them to appear to grow faster due to the abundance of nutrients and low light-limiting photosynthetic output to provide to that fungal partner. These mycorrhizae are also going to be found in the rhizoids and roots of gametophytes and sporophytes. With the gametophytes, they're going to enter the rhizoids in response to hormones that are released by the plants. They received increased protection against drought and nutrients and sugars from the mycorrhizal association. Now with the sporophytes, it does not inherit the mycorrhizal association from the gametophyte, but it has to acquire it from a substrate once its roots start to develop. Studies suggest that mycorrhizal colonization occurs better in the nutrient-deficient substrates and in thick, fleshy roots. For reproduction, they're going to have both asexual and sexual reproduction. With asexual reproduction, you have apogamy. That's going to be the development of a sporophyte from a gametophyte without fertilization. You have vegetative propagation. That's going to involve the sporophyte and gametophyte producing vegetative gemma that are going to detach and develop into a new plant. And you have fragmentation, which we've talked about before as well. That's where the sporophyte rhizome can break off and produce a new plant. Sexual reproduction is going to involve an alternation of generations life cycle. So you're going to have a 5-step process here. The adult diploid sporophyte is going to produce haploid spores by meiosis within the sporangia. Once those spores have reached maturity, the sporangia is going to open and those spores are going to be dispersed by the wind. After landing on moist, sustainable substrates, the spore germinates and begins to mitotically grow and develop into a mature haploid gametophyte bearing reproductive gametangia and archegonium and theridium, where the mitosis produces eggs and flagellated sperm. In the presence of water, the antheridium is going to release flagellated sperm, which are going to swim to the archegonium to fertilize the egg, producing a diploid sporophyte zygote. Development of the diploid zygote is going to continue by mitosis within the archegonium until the first sporophyte structure begins to differentiate, such as leaves are going to be present, and you're going to have root-producing rhizomes. Upon gaining autonomy, the diploid sporophyte is going to maintain its development towards a dominant, mature adult plant. As far as nutrition, these are going to be photoautotrophs. They're going to require water and minerals from the environment to assist in plant growth and metabolic function, but they acquire these nutrients in a variety of different ways. We have terrestrial and aquatic pteridophytes, which are going to use root systems to absorb nutrients, but aquatic ferns are going to belong to the zola, and they're going to fulfill their nitrogen requirement utilizing a mutualistic association with nitrifying cyanobacteria. Here we see the nostoc, which are going to help the azolla in order to gain their nitrogen that they need. Epiphytic pteridophytes are going to live on the surface of tree trunks and branches where they're going to be able to trap and absorb nutrients from the crown humus or from rainwater using specialized leaves or modified root systems. We have the Lichenopteris that's going to help with the epiphytic fern species, and that's going to receive nutrients by means of mutualistic ants that are going to typically reside within their shoots and rhizomes. So in this picture here, you're going to see the Lichenopteris. Now the pteridophytes are going to be seedless tracheophytes with nearly 12,000 diverse species. They're going to be classified into 3 separate divisions. First, you have your Lycophyta. They comprise about 1,300 species of quillworts, club mosses, and spike mosses. With your quillworts, they're going to have deep, penetrating, hollow root systems, and they're going to have kind of like a simple stem with a microphyllus with leaves, and then they're going to have heterospores on them. With the club mosses, they're going to have a spirally arranged microphyllus leaves that are going to be located on upright and creeping stems, and they're going to produce the homospores and the sporangium that are going to be found among the leaves or in the club-like strobili that are protected by the saprophyls. With the spike mosses, they're going to be heterosporous plants with sac-like microphyls. Each is going to have a distinctive lugule on their upper surface. Next we have the Arthrophyta. These are going to be your horsetails, and then your Monolithphyta. These are going to be your whisk ferns and ferns, and we're going to talk about these more in a minute. So let's go back to the Lycophyta. Looking at the quillworts. Within the quillworts, there's nearly 200 species. These are going to be found in the terrestrial, amphibious, and aquatic habitats. So you find them usually on land, or they're usually near water sources. They're characterized, we said, like the bulb-like rhizome that gives rise to deep penetrating hollow root systems. Like we said also, you can see here they're going to have this simple stem, which is going to be called a corm, and it's going to have that microphyllus leaves. And so you have these long, thin, filamentous looking like leaves. They have heterospores, which you see here, and they're going to be small male monolete and large female trilete spores. Coming down with the club mosses, there's going to be about 400 species of club mosses. These are going to be found in forests from the tropics to the Arctic. They're characterized with those rhizomes that produce shallow branching root systems that you're going to see in this picture. They have upright or creeping chlorophyllous stems, and that's going to be like spiraling and arranged in the microphyllus leaves. They're produced the trilate homospores in the sporangium, and they're going to be found among their leaves of like club-like strobili, and they're protected by these sporophylls that we talked about. And with the next one, the spike mosses, there's nearly 750 species of these. They're found worldwide, with many distributed in the tropics. They're characterized by roots that arrive from branching stems. These stems will grow upright or along the ground. They have simple, scale-like microphyllus, and they're going to be distinctive with a flap-like tissue called a ligule, and that's going to be on the upper surface, as you see. They're going to produce heterospores. They're going to have small male trilete and large female trilete spores. And these are going to be protected by the strobili sporophylls. Now going into the Arthrophyta. These are going to be your horsetails. There's roughly about 20 species. They're going to grow in or near moist, marshy habitats around the world, and they're characterized by deep horizontal rhizomes that are going to produce true roots that you'll see in this picture. They have hollow, rigid stems that are going to be comprised of silica, making it abrasive and very rigid. They have whorls of microphyllus leaves. They're going to have homosporous, and sporangia are going to be arranged in the strobili. And so you can see that here in this picture. The Monophyllida is going to be the whisk ferns, and this is going to be roughly 12 species. They're subtropic plants that are comprised primarily of epiphytic and terrestrial species. These are going to be characterized by the inability to produce roots, but they can anchor themselves by horizontal rhizomes that produce rhizoids. Their highly branched, aerial stems are going to give them a distinct broom-like appearance. Leaves are going to be highly reduced, they're going to lack vascular tissue, and in some species they appear as small scale-like projections along the stem. And they're also going to be what we refer to as the homosporous again. And so here you can see these in both pictures. So with the ferns, these are going to be the largest number of pterophytes with nearly 11,000 aquatic and terrestrial species. They're endemic to the tropics, but found across many different habitats around the world, including deserts and the Arctic. They're going to be characterized by true fibrous root systems that are derived from underground rhizomes. These stems are going to range from creeping to long and thick, and they're going to be found in tree ferns. Broad megaphyllous fronds that are going to begin as fiddleheads and often are going to be used as sporophylls are going to be clusters of sori that are going to be arranged underneath. And so over to the left, you're going to see these fiddleheads that they have taken up. In the middle and to the right, you're going to see the fronds. So the fronds are going to be these long leaves. That's what it's going to develop into from those fiddleheads. Let's go back for a second. So underneath these fronds, you're going to be able to see these sori. The sori are going to be arranged in clusters. And when you look at them, if you can see them, the little dots that are going to be on the mature sporophyte that you see in the picture below, those little dots, those are going to be the sori that are going to contain the spores. So here you see sorus, and then you see that the spores are going to be underneath them. Okay, so most species are going to be homosporous. Although these spores are going to produce prothallus in some species, they're going to be different variations, and you're going to have morphologies of gametophytes that are going to be found in other fern species. Ecological benefits. They're going to help to maintain water balance, participate in nutrient recycling, and provide microhabitats for other organisms in habitats that they occupy. They're considered ecological indicators by most scientists due to their sensitivity to different forms of environmental change, such as habitat destruction, degradation, climate change, and pollution. So if places that usually have ferns see their ferns starting to die out, then they know that there's some type of pollution or something going on that is damaging that ecosystem. Human benefits. They can make and maintain household items as a primary food source. So you can have them around the house or you can actually eat them. Medical benefits is that they're used for thousands of years in traditional and herbal medicines that make things such as tinctures or ointments to treat, cure, or heal a wide range of illnesses and ailments such as arthritis, diabetes, and to help heal wounds. Modern science has discovered beneficial medicinal values of specific compounds produced by the pterophytes that have displayed antiviral, anti-cancer, and antifungal activities. And they show promise in creating effective antibiotics and in treating certain diseases such as Alzheimer's and diabetes. Some agricultural benefits. The efficacy of compounds that are produced by the pterophytes against agricultural pests are going to be considered as an alternative for the use of synthetic pesticides. So these might be a way that we're able to deter pests without putting different types of chemicals, let's say, into our food or into our soil and water sources. The azolla has become an important fern that's going to be used by rice and livestock farmers as a biofertilizer, and its supplemental nutrients to improve their agricultural production and output. Horticultural benefits. It's going to be used in gardens. It's used as in pots. It could be used as a floral decoration to display the aesthetic value. That's going to finish us up for chapter 16, or 15, sorry. See you back for chapter 16.